Hello, Feather. Nice to have you here. I will allow my panelist colleagues to answer the questions as I'm realizing that all of my screen is being shared right now. In any case, I think I am going to go ahead and get started. Thank you, everyone, for taking the time to be here this afternoon. My name is Zachary Flegel. I'm the Technical Sales Specialist at Nexus Scientific. I'm here today to present a webinar on label-free, high-resolution holotomography in partnership with the company Tomocube. Today, we're actually going to be discussing both their imaging technique and highlighting the conference that we will be at next week. We will be participating at the AIM conference. Actually, sorry about this. Let me move things to the bottom. We will be attending the AIM conference January 24th through 26th. So just want to thank all the sponsors, everyone helping to make this event possible. This is sort of the pre-conference webinar event for this. That being said, still a good opportunity to learn about the system if you can't make the conference. In addition to that, there will be a live showcasing of the HTX, that is Tomocube's latest holotomography system. The time lapse on the previous slide was taken with that system. It will be at the Berkeley site throughout the conference and for a number of days afterward. The date will be corrected. It's actually going to be January 20th through February 4th. So for any of those that are in the area interested going to the concert, please follow up. Towards the end of this presentation, we'll actually have some sign-up links for you to go to, and you can reach out to one of us and schedule your live demonstration, again, either before, during, or after the conference. During this presentation, there is an option for people to ask questions in the chat. It won't be any live feedback, but there is a Q&A portion of the presentation, around 30 minutes or so, where we'll open up the floor, and I'll have some colleagues from Tomocube open to discuss any of the questions you might have that are written in the Q&A. So as questions come up, please don't hesitate to write them in there, and we will answer them as they go. So the first thing I'm going to do here is actually ask a poll. I believe you should be getting a poll question, sort of a fun question to sort of start things off and really interact. We have a number of these throughout the presentation, but the first one is, which social media are we using most often for scientific posts? I do think I know the answer. I'll personally give my answer, and LinkedIn seems to be the number one, at least for me and my colleagues. But so has been said, the scientific community does have a Twitter presence, though I have not delved into it myself. I'm going to stop the poll now. I believe I can share the results with everybody. LinkedIn was, the prime answer there. Did expect that one, but curious to know what the 4 out of 12, what the other ones other than what was mentioned are. But we can get to that. Anyway, yeah, just wanted to get everybody familiar with the interface there. There will be a couple of other poll questions coming up during the presentation. Before we start, I want to differentiate myself a little bit. I mentioned I'm with a company called Nexus Scientific. We are a small biodeck distribution company based in Boston, Massachusetts. We work with not only Tomocube, but a number of other overseas companies specializing in live cell science research. And we act as U.S. distributors, service, and technical specialists, assisting them in bringing their sciences and technology to the U.S. What is holotomography is going to be the question of the webinar, and why is it unique, and how is Tomocube doing what they're able to do? Essentially, it's a form of label-free high-resolution imaging. Unlike most techniques where they're measuring either a phase shift delay or a fluorophore excitation, we're actually taking quantitative measurements of the physical properties of the cell, namely the refractive index distribution across a volume. We are getting 3D information as well. And as you'll see, based on the light techniques, we're able to sort of go a level beyond your other gentle techniques, such as bright field and phase contrast, and really get the resolution that we need for some of these experiments. here we have a panel of images. I'm sure we're familiar with a lot of the forms of microscopy that we're using to do live cell imaging. There are really two essential domains when it comes to this fluorescent microscopy and all its variables, from confocal, light sheet, spinning disk, tons of methods. You do get benefits with fluorescence, of course. You have high specificity. We know exactly what we're targeting. We get usually good signal to noise. But the downsides are, of course, we do have to somehow stain the sample or get it to express a certain fluorophore, and then we have to excite it with light, which is both going to bleach the fluorophore and hurt the sample itself. On the phase contrast DIC side, we are still having a gentle imaging technique. Things are in a much more natural state, but we don't get the resolution or the detail, the quantitative information we need in a lot of the questions we're trying to have answered. With holotomography, we get the best of both worlds. We're still getting very high resolution. We're getting quantitative data, but this technique isn't going to cause any damage to the cell. We don't have to do any staining, so we never have to compromise between, how fast can we image or how long can we image it for. Another note about both of the systems that we'll talk about today and throughout the Tomocube technologies, while of course the label-free imaging is our main goal, we are able to still correlate 3D fluorescence imaging to combine and complement the data. What are we actually measuring? What is holography itself? We're actually, when we pass a light through a transparent specimen, this is a transmitted light technique, the light is going to be delayed by the dry mass or the refractive index and the thickness of the sample as it goes through, as that beam of light goes through your sample. In most techniques, you can't differentiate these two properties without some sort of way to get three-dimensionality. However, if you come from multiple angles and involve tomography in this concept, you can actually differentiate the refractive index and the thickness of the sample, and we can measure both. So this is what the light pathing looks like. We have a dual beam technique in one of the systems that we use here. One beam is going to act as the reference beam. It just hits the detector and goes onto the camera. The key point is that we know everything there is to know about this beam. We know the pattern, we know the timing, and then we can compare that to a beam that then goes through the sample. It's obviously going to be delayed by both the thickness and the refractive index of those components. And then we do that not once, but we illuminate the sample from multiple angles. So we use what's called a digital micromirror device to actually project the light at a 45-degree angle around multiple points throughout the sample. So it's a combination of both measuring that phase shift delay and comparing it to the refraction or the reference beam and illuminating it at multiple angles with this digital micromirror device. There are two forms of holotomography. The first one that I just discussed was a conventional holotomographic system, which the first iteration of the Tomocube systems use. It is a coherent light source system. The system is called the HT2H. It does have the advantages of having a faster acquisition, a little bit higher resolution, and better axial resolution. But the system I really want to talk about today is a version of low coherence holotomography. That'll be the HTX1 that took some of the earlier images that you saw in this presentation. It does have a number of benefits that I'll go over in more detail. The primary ones are that we have a much higher signal to noise ratio in the fact that we can pretty much eliminate speckle noise. There's no background calibration step. It's a much more streamlined instrument, and it's compatible with multi-wall plates. Both of these techniques, in some way, the light paths are a little different for each one just due to the way they are built, but both of them are essentially relying on this holotomography type pattern. It's very similar. A good way to think about it is of a CT scan. We're, instead of going around the sample with an X-ray motor and using a human specimen, we're actually looking at cells, measuring the refractive index, and using a laser or an LED, depending on which one of those two methods of holotomography it actually was. So all in all, combine the two concepts, holography, measuring that phase shift delay of the light as it goes through the sample, getting the RI and the thickness, and then being able to differentiate the RI and the thickness by capturing holograms at multiple angles. That gives us 3D information in a very short amount of time. So all of the data that you'll see in this presentation, even the 2D sort of videos that you saw at the beginning, those are maximum intensity Z projections. We do get all of the Z stacks, and we are capable of doing 3D volume reconstructions of all of this as well. Of course, it is quantitative rather than 2D methods. So your other gentle techniques, bright field, phase, and DIC, where you can really only get things like area, confluency, cell counting, 2D parameters. With holotomography, because we're getting 3D data, refractive index actually correlates to dry mass. We can get concentration from there as well. And we get the 3D versions of those measurements, so volume, sphericity, and surface area. All right, now we have one more poll question coming up. And this is about, sort of just to get a gauge on everyone's imaging techniques that they're currently using to image live cells. And this can, obviously there are multiple ways to do this, but sort of the most common, what's giving you the most results at the moment, just to see where everybody is. All right, I'm stopping the poll in just a couple of seconds here. Sharing the results. And as I thought, fluorescence microscopy in some way, shape, or form is the dominant method to image live cells. It is really the gold standard of live cell imaging right now. Specificity, we can get very high resolution. But again, it does have some downsides. And that's what really is going to lead us into this next proportion of the presentation. Oops. Which are why holotomography, what are the advantages over the other types of imaging? So here are the 5 main reasons, and I'm going to follow them up with a couple of images, a couple of examples for each showcasing, but just to go over them, no labeling, that's very important for both sensitive and high temporal resolution. Low phototoxicity, again, great for sensitive samples or sensitive processes you might have to monitor either very quickly or over a long period of time. We can get 3D information way faster than any sort of confocal Z scanning, just because of the fact that we're taking the angles rather than moving an objective vertically up or down and doing any sort of traditional Z stacking. We're getting higher resolution. That's what's beyond the diffraction limit for this case, because we're oversampling using that tomography. And it's quantitative data with consistent image intensity. We don't have to rely on, or we don't have to worry about any photo bleaching or decreasing signal over time. The first one, no labeling. While we don't have the specificity that fluorescence does, there is an advantage to not having specificity in that we get to see everything that we do get to see. So this both helps in addition to the fact that we can see many organelles in one picture, but in cases where if we're just doing fluorescence, some cells might not uptake the dye properly, and we might miss a lot of the events that are going around, especially in the ecosystem of this cell. So what can we actually see on a good day with holotomography? What sort of organelle ecosystems? We have lipid droplets and mitochondria. So there's your metabolic processes. Lipid droplets obviously have a high refractive index and are by large the easiest thing to visualize with this type of modality. Usually getting good detail around the nucleus, we can see the nucleoli. If we were here for that first video, we saw the chromosomes condense and line up during mitosis. Good detail around the envelope as well, so we can see, especially in combination with fluorescence, any sort of trafficking of proteins or anything within the nucleus. Or in this case, we can see lipid droplets in the nucleus as well. And the last thing I'll mention is great detail around the membrane itself. So any sort of uptake, any cell-to-cell interactions, any sort of trafficking experiments are all great applications, especially in combination with the fact that we can do this at a very high acquisition rate for a long time. Low phototoxicity, very important for your sensitive samples or for very long processes. So in this case, we were able to image cells for a multitude of days. These are pre-adipocyte cells. We can see the lipid droplets growing in size over the course of the experiment. And on the right, unfortunately, not a video, just an image of some human embryonic stem cells. And the highlighting point here, again, is that anytime you have a sensitive process or a sensitive cell line, the last thing you want to do is have to stain them and then have to excite them with fluorophores while you're doing your experiment. That's always going to be a confounding variable. And then, of course, when you're doing those sorts of acquisitions, you have to worry about how often you're actually acquiring those images. All of this that you see here is in 3D. And we're capturing images depending on which system, anywhere between half a second and around 6 seconds, maximum acquisition time. And every time we're taking it, we're getting 96 full Z stacks. So here we can see just a couple of different Z layers in this case. But what we can also do, which I think is much more visually appealing, is do the 3D reconstructions of these as well. So this is a digital staining with the pseudo colors representing different refractive indices. And then we have another image of cell death here on the right. And very powerful, not only to, of course, have those 2D images, but then to be able to replicate this in 3D. And all of this is label free. You can see sort of the different compartments we can visualize and imagine for every frame, we're doing this in just a couple of seconds, as opposed to any sort of confocal Z stacking just to build out that stack with, with a couple of colors. Not only is it going to take more time, but it's going to cause much more damage to the cell just with the number of exposures. I mentioned we do get a high temporal resolution, higher than that actually, that the hardware of both systems would suggest. We're using 1.2 NA objectives and I think 0.9 objectives for either system, but in both cases, we're still able to get around 110 resolution for one and 160 resolution nanometer X and Y resolution for another system. And this is due to the fact that we're oversampling essentially in all 3 dimensions when we are making the tomograms. So when we're revolving around the sample, or not revolving, when we're projecting the light around the sample, we have many different overlapping images that are being aggregated to form this traditional image. So we get great detail, great measurements. And here we have the refractive index bar at the bottom left. Lastly, the fact that we are getting this type of quantitative data and that we don't have to worry about losing any sort of signal makes this great for applications where we really need fast temporal dynamics, and then especially, of course, for an extended period. So lipid droplets, I mentioned, are a great application for any sort of holotomography because they always show up very clearly in the holotomography due to their very high refractive index. And here we just have a panel of cells imaged, and you see the photo bleaching from the lipid dye on the bottom panel compared to just the holotomography and the a version where the lipids are segmented a little bit more clearer. And you can see that, after 36 minutes, the image quality doesn't degrade at all. And that would be the same if we had taken, 20 images in between each one of these time points. We can image for virtually as fast as each system can go for as long as we want, because we're not causing any damage or using any fluorophores. Here is another example of that. And these differentiated adipocytes were actually imaged for, I think, a period of 72 days. I think my screen sharing bar is actually blocking things out, but these were imaged for multiple days, and you can see that the signal does not change over the course of those days. With reference to photo bleaching or anything like that, obviously the signal will change depending on certain processes going on, but I think we get the idea. All right, I will stop briefly for our next poll question. Maybe I won't. I'm having some technical difficulties on my end launching this last question. I do apologize. I don't think I'll waste any more time trying to find out. It was kind of a gag question. The question was, what was the most obvious organelle, organelle that was visualizable on the last image? And I'm pretty sure the obvious answer should have been lipid droplets. All right, up to this point, I haven't really talked about fluorescence, but I did mention at the beginning of the presentation that we still are able to do these correlative fluorescence microscopy studies. So we can do fluorescence in 3D. So we still have the capability of doing wide field fluorescence and Z-stacking and correlating the 3D fluorescence with the label-free 3D data. So this is just a maximum intensity projection. We have lipid eye and hoist versus the RI maximum intensity projection. This is obviously important because we want to, while we want to do everything in label free, realistically, we do have to be scientists and say, well, you know, how do we know these are lipid droplets in this case and not lysosomes? We need a better way to figure this out. Even though now with holotomography, we've realized that the refractive index differences between those two organelles are substantial. We still need to do this correlation in some instances to make sure what we are looking at is what we're looking at, especially when it comes to other organelles, so mitochondria, things like that. Want to make sure that these objects are what they are. And of course, we want to be able to track anything that can't be visualized in the RI itself. So any kind of protein that we want to see trafficking, we can also monitor in 3D. Of course, it is in 3D, so we want to visualize it in 3D as well. Here we have both the fluorescence in 3D and then an overlay of the fluorescence with the holotomography. It does get a little bit crowded once you have 5 or 6 colors in there, but it gives you a good representation of what we can do. So the blue and the sort of light gray there is the refractive index distribution, and we can overlay that with this 3D image here. I mentioned Tomocube currently has 2 systems, the HT2H and the HTX1. One of them features, the HT2H features the coherent holotomography, while the HTX1 features the incoherent holotomography. And these systems aren't, there is a lot of overlap in terms of their application, but there are applications that suits one or the other separately. The HT2H does have those faster dynamics and a little bit higher of a resolution. So depending on if you need high resolution, might be a better fit. So, you know, things like lipid-liquid phase separation, microorganisms, membrane dynamics, if you need really high acquisition rates, those are better. However, the HTX1, the newer system, the one that will actually be at Berkeley, has many of the advantages and even more with this low coherence holotomography technique. Just a couple of the ones I want to highlight in the next few slides is that because we've altered the technique a little bit, we made it compatible with multi-wall plates. There's a laser-based autofocus We can work with highly confluent samples. A longer working distance allows us to actually penetrate deeper in Z, so we can work with thicker samples. We still have the correlative fluorescence imaging, of course, and the hardware is a little bit different as well. This has a 40x objective, a little bit lower end or lower NA compared to the predecessor microscope, still getting 160 nanometer X and Y resolution, still a high resolution system, but getting it at 40x, so a much higher field of view than its predecessor as well. All of this fits inside of this refrigerator-like device. There's a stage top incubator in there as well to control for environmental conditions. Motorized stage, going to move between wells, do tile stitching, and everything a modern microscope system should have. I do want to mention our current situation with the multi-well plates. Right now, we're going up to, or we are compatible with 24-well plates with aspirations to move a bit higher. I like to highlight this just to mention that there is a challenge when it comes to this type of imaging and scaling it up to multi-well plates, because we have to hit the sample at an angle that does limit the size of the well itself that we're able to use, and correspondingly the angle that we have to illuminate at. And that has really been the challenge to sort of adapt the holotomography technique to multi-well plates. Right now we're up to a 24, and we're hoping to increase things from there, but 24 is still better than 1, especially when you combine it with the fact that we're getting larger images and can tile stitch. Moving into some of these HTX images, a couple of things to highlight. So here we have the high contrast holotomography example. Great detail around the background. I believe this was the Hep B 3B cells from earlier. So we can see all of these organelle ecosystems, mitochondria, lipid droplets, nuclei, even some structures within the nucleus itself. And all of the images come out like this. This is, we haven't adjusted the data in any significant format here. This is how the maximum intensity projections just come out. Extremely clean images. As far as throughput, we are capable with this HTX of scanning multiple millimeters by millimeters at this 165 by 165 sort of 40x field of view. Here are those adipocytes again. In this case, you can just think about sacrificing the temporal resolution for getting a greater number of cells, but still all at this high resolution. Something that is unique about the HTX compared to any other holotomography system on the market is this larger field of view with a longer working distance. The longer working distance does allow us to penetrate deeper into the samples, and we have been able to look at some small organoids and tissue samples. So this is actually a lung organoid. You can see us looking through the different Z layers here. And we also have a visualization of this, highlighting some of the different organelle ecosystems we can see as we scroll through the Z-plane of this lung organoid. And you could just imagine trying to, any sort of label-free method, sort of phase or GIC. Obviously, you're not getting, subcellular data with those techniques, especially not in an organoid. They're pretty much just looking at black dots from a certain point. And here we're able to resolve different subcellular structures in different layers of the organoid itself. Here, a good image of sort of the tile stitching, as well as just the resolution. It's a cool video I like to show because it's really an application that we don't usually see in high resolution. Usually with any sort of wound healing assay, we are looking at a much lower magnification, much lower resolution, which is valid in a lot of sense if we're just looking at sort of population level metrics, how fast is the wound closing. But here you can really see, how are the cells moving? You can start to think of a few more questions to ask when you're really looking at the closure at such a high resolution, rather than just, how fast is my gap closing? Here are a couple more images just of that 3D lung organoid. I think those slides were backwards, unfortunately, but just highlighting the various events that we could see within each different layer or various portions within that layer. So here, we have the ciliated surface. There's that mucosal layer we saw with the cilia penetrating through. Even have a cell division that we could clearly spot in one of the layers, which was pretty cool. The next application I want to briefly cover is lipid droplet analysis. I like this one because of course, it's the easiest one for holotomography to perform. There's a few published papers on it already, and that it's just the most good-looking presentation or most good-looking data set that we can produce, just because the images are so good when it comes to the technique. So here we have a couple of different data sets looking at morphological changes and lipid droplet accumulation in foam cells. And then at the bottom here, we also have quantification of lipid droplets in response to oleic acid. So looking at both the number of lipid droplets and the size of the lipid droplets, and the clear differentiation between the control and the oleic acid group. And then, of course, having the holotomography to really visualize that, and not just in this RI map in 2D, but in a 3D space as well. I mentioned it's extremely easy for this to quantify. One of the things that the Tomocube software does well is this automatic lipid thresholding. Currently, the software is where the bulk of the work is heading for Tomocube. Looking forward to having a lot more analyses like this coming on the way. But right now, where the system is shining is obviously looking at these lipid droplets, doing the segmentation based on the RI, and then calculating the volume and the concentration, and of course the count of those lipid droplets, hoping to then extend this sort of logic to see what other analyses can be done with the system. The last application I will close with is mitosis. I started with the mitosis video. I will end with the mitosis video. Anytime you can visualize a process like this, that obviously cells want to be dividing when they're healthy, or when they consider themselves healthy. I'm just thinking about cancer cells now, quote healthy. Anytime we can visualize that without adding any native substances, this is obviously going to be even more relevant for primary cells. Anytime we derive samples, we don't want to introduce any sort of those confounding variables. But here we can see very clearly all of the different phases of mitosis. We can see the label-free condensation of the chromosomes, and we can even see good detail around the mid-body and the daughter cells as they're dividing. All right, here is that last video of mitosis. This was the same video played at the beginning for those of you who showed up a little bit later. This is one of my favorite videos. I think it has great detail. It was taken with the HTX. You can clearly see mitosis and the division, but not only that, I think the digital zoom allows you to really get an idea of the type of detail we have along the membranes of both these cells, but also in the dividing cell. We can see again the mid-body there. All right. So that is the webinar. I am going to transfer over shortly to the Q&A. Before I do, I just want to say thank you, everyone, for taking the time to attend. Thank you to the AIM conference organization team at Berkeley for assisting to set this up, as well as the conference. I hope to see many of you and look forward to seeing you at the AIM Berkeley conference next week. And all right, yeah, I will open the Q&A. I will also allow some of the Tomocube team to come chat, and we can answer your questions from there.
Hi, Zach, can you hear me?
Yes, I can hear you, DJ.
Okay, Thank you for the excellent presentation. Yeah, I was just answering some of the questions. To those everyone here, hello, my name is DJ Shin, and I'm the Sales and Application Specialist from Tomocube. Yeah, any questions that you may have, I am looking at the Q&A section, and I will be answering them one by one. The last one that I was looking at was how to quantitate the image or cell size, activity, migration, etc., without fluorescence labeling. As Zach may have mentioned, our imaging is based on the refractive index of the cells. So the refractive index is actually directly proportional to many biophysical parameters such as volume, cell volume, dry mass, and protein concentration. So as we image the cells, we acquire the 3D voxels, and we can quantitate the protein concentration and dry mass of each voxel that we can see. And do you have the slides or other documents to share by email? We will be providing this presentation by demand for those of you who have missed it or may want to watch it again. The link for that will be emailed to you separately. Oh, and I am seeing another one. To image internalization of nanoparticles, the refractive index is significantly different to distinguish. This one is a bit trickier. So we do have some experience imaging nanoparticles such as silica dioxide and gold nanoparticles, but it does depend on the thickness and refractive index of the nanoparticles. For example, we found that the refractive index for gold was much higher than the surrounding area of the cell, and also including the video that Zach showed of the 3D internalization of the nanoplastics inside the alveolar cell. So in that case, the refractive index of the nanoplastics was distinguishably higher than the rest of the cell. So in those cases, we are able to image those nanoparticles quite clearly. But it does depend on the thickness or refractive index difference between the nanoparticle and the cell, I do have to mention.
Yeah, thank you again, everybody. For those more specific questions about the event in Berkeley, I think Suman indeed did a good job getting to most of those. And in just a moment here, I'm going to actually share in the chat with a link that will register for various times, again, both before and after the AIM conference to either come by and see the system or perhaps even test out some samples. Okay, in the chat, you should there should be now a link to sign up for that visit. Do have one more question that just got brought up that I will answer. Is it possible to separate or see a specific RI in an image specific to fluorescent channels? Excuse me. The answer is yes. That's actually how I don't, if you saw those 3D representations, those were actually ranges of refractive indices that were segmented. So the idea there was, we essentially set the software to, say, take this range of refractive index values and this range of an index gradient, which is the other variable we measure, or we pick them out on the screen and it'll tell us the range and we can adjust from there. But yeah, we can pull out specific refractive indices. That's how we do that label-free segmentation. All right, I think we are coming up on 2:45 here, which I believe, to my knowledge, is the endpoint. Panelists, correct me if I'm wrong. If not, I would like to again just thank everyone for attending and thank you for your questions. If there are any last-minute questions, please don't hesitate to reach out, or if there was any unclear answer, feel free to post again. Feel free to visit the Calendly link that we put in the chat list to register for those events. For the rest who aren't able to attend the conference but are still interested, we will be following up with the attendees of this webinar through email to discuss any sort of interest further. If you have interest in the Tomocube or want to learn more about Nexus, we can surely assist you from there.